We have talked about the structure and function of cells, but a necessary next question to ask would be, where do cells come from? And where did the first cell come from? The field of biology has a few answers to these questions, some of which are theories, which are a very big deal. In addition, a lot of research is being done to further understand the origins of life on our planet. Let's start with the simple question of where do cells come from, as in the cells that we are made up of and other cells that we see and eat on a daily basis. Based on how cells work, which we have derived into a set of parameters called cell theory, cells can only be formed from an already existing cell. The process of how this works can look different, but cells are able to grow and divide and create new cells in the process. We can refer to the original cell as the mother cell and the newly created cells as the daughter cells. This is well documented and manifests in all living organisms, whether they are dividing by mitosis, binary fission, or some other means. We will go into the details of cell division in section 1.6. Now, the question that always pops up, if cells are only created from pre-existing cells, where did the first cell come from? Clearly, we can't make a cell if there are no cells around, right? The answer? The first cell must have risen from non-living material. This theory is called abiogenesis. It starts with the spontaneous creation of simple, non-living organic molecules. These molecules, driven by the laws of physics and chemistry, end up assembling into larger, more complex chains or polymers. And just like if you put crystals in a solution, these more complex molecules begin to self-replicate. This process of replication started to build memory within the context of these non-living molecules. From there, these molecules became packaged into membranes, which we call protobionts, or early abiotic cell formations. These then continued to build complexity and replication abilities and were molded into the earliest version of prokaryotic life on our planet. Scientists have conducted experiments to test this theory, focusing on the concept of creating molecular complexity within a system that doesn't have any. This non-living synthesis of simple organic molecules was demonstrated by the Miller-Urey experiment. To sum this up, they took basic chemicals that would have been found on our planet prior to the origin of life and recreated the environmental conditions. With the correct chemistry and also the right conditions, they were able to synthesize organic molecules from components that were chemically unique. A pretty awesome experiment if you ask me. Experimental data has shown that in the early environments of Earth's history, complex molecules could have been created, which over billions of years could have led to the complexity of life that we see on our planet today. But what about making new life today from non-living organic molecules? Shouldn't we be able to make organisms spontaneously if we have the correct components? The answer is we can't. Based on what we know about cell theory, we have come to the understanding that all cells must come from pre-existing cells. We can't simply make new cells from just their components, even though that seemed to be the case for abiogenesis. The main difference here is that the current conditions of our planet are nowhere near what they were billions of years ago, which helped spark the creation of that first ancestral cell. In the 1860s, Louis Pasteur figured out that heating liquids at high temperatures and then slowly cooling them would eliminate bacteria inside the liquid. We call this process pasteurization. An experiment he ran subsequently helped support the concept of biogenesis, or that cells can only be created from pre-existing cells. Here's how it worked. He started with a broth solution that contained microorganisms within it. The solution was boiled, which killed all of the microorganisms, creating a sterile environment. Now at this point, the microorganisms were killed, but that does not mean their molecular components disappeared. Pieces of those destroyed microorganisms were still in the solution. The solution was then cooled down. When the solution was sealed and the environment still sterile, there was no additional growth in the flask. So even though the broth had the proper components needed to build life, nothing happened. No life was built. No new cells were created or spontaneously generated from this solution. But when the flask was opened up to the exposed outside environment, bacterial growth would shortly follow. This was because opening the flask to the non-sterile environment meant some bacteria would make their way into the flask. consuming the contents in the broth and reproducing. Again, this shows that once cells enter the solution, you will get more cell growth, solidifying the idea that cells can only arise from pre-existing cells. As we know from the previous slides, life started off on our planet with very simple single-celled organisms. But when we look around our planet today, we see a lot of complexity. How did this complexity come to be? How did the planet start with prokaryotic cells and have eukaryotic cells develop alongside them? We can answer this question with the theory of endosymbiosis. Endosymbiotic theory describes how this complexity was achieved through the creation of symbionts, which is when a cell lives inside of another cell and both mutually benefit from the interaction. This works when a large cell engulfs a smaller one, but does not break it down. It continues to survive inside the large cell, in which it receives some protection while the larger cell benefits from obtaining molecules it produces, like ATP. We have strong evidence that supports this theory, like the fact that some organelles, like the chloroplasts and mitochondria, have two separate membranes, prokaryotic 70S ribosomes, and they have their own separate DNA.